Hello, grade A students. In this session, we're going to continue with our enzymes in the chapter of digestion. So in the chapter of digestion, we talk about the enzymes. We said that the enzymes, they are the agent of digestion. They are the biological scissors that are used to cut or to break down the food into smaller pieces. So what are the conditions needed for the proper functioning of the enzyme? What do the enzymes need in order to work
Good
properly?
morning, Stan.
So here, interpret the results of experiments shown, deduce the enzymatic property verified in each of the performed experiments. So here we have in this experiment, we have two test tubes. Both tubes, they contain fresh saliva, meaning that fresh saliva, meaning that it contains the enzyme salivary amylase. So both of them, they contain fresh saliva. In tube A, we have cornstarch. In tube B, we have albumin of an egg. So here in tube A, we have cooked starch. So salivary amylase, it works on starch to give maltose at temperature 37 degrees Celsius. However, in tube B, we have fresh saliva with albumin. Albumin, as we said before, it's a protein. Albumin of an egg, it's a protein. So this protein is what? So this protein is the one that needs another enzyme other than salivary amylase. So in experiment in document B, we have the results obtained. The results obtained in document A, we have Fehling test. It gives brick red precipitate. This means that we have reducing sugar. So this reducing sugar is obtained from the digestion of starch into what? Into the maltose, which is the reducing sugar. From here, you have to review the tests. The tests are very important to know them and also in order to know the different foods obtained. So here, reducing sugar, so we have the brick red precipitate. So the starch is digested into reducing sugar. So this is digestion. However, in tube B, after 1 hour, so here it can say 7, and here it becomes 8. So here after 1 hour, the violet test, it gives a violet color with QP. So violet test is for what? It's for proteins and slash or polypeptides. So proteins and slash or polypeptides. So either proteins or polypeptides. So here we have positive result. So here peptides or proteins, they still persist. They are present here and they are not digested. So digestion occurs only in tube A and not in tube B. So whenever I ask for interpret is analyze plus indication. Not like one of you or some of you they did in the quiz. Whenever I ask about interpret, it's analyze plus indication. Without an indication, this means that you are not doing interpret, you are doing analyze. So this time, I accept I gave you half of the mark for this part. Next time, if you did analyze instead of interpret, so I'm going to remove all the mark. So this is what I'm going to do in grade 9 next year. So you have to know that interpret, it's analyze plus indication. Those who did only analysis, meaning that they don't know what the meaning of interpret. So next time I will drop the question. So you will not get any mark for the question. So after adding fresh saliva to the quick starch in tube A, the result is brick red precipitate with Fehling. This indicates the presence of reducing sugar. What? Obtained from the transformation or the digestion of starch in the presence of fresh saliva. However, after adding fresh saliva to albumin in tube B, the result is we have violet color with violet test. What does this indicate? This indicates the presence of proteins and or polypeptides, and they are not transformed by the fresh saliva. Here, the first property. Every enzyme works or acts on a specific substrate. Second, of the pH. Pepsin is known to work in an acidic medium. Salivary amylase works in a neutral medium. However, trypsin in basic medium. So here we have acidic, neutral, and basic. So here pepsin, it works best at the acidic medium, while salivary amylase it works best at around neutral medium, which is the around the neutral medium, which is 6.5 to 7. However, the trypsin enzyme, it works best at basic medium where the pH is greater than 7. So here the enzymatic property reduced. Every enzyme works on a specific pH medium. So I repeat, then now we have two different properties for the enzyme. Every enzyme works on a specific substrate, and second one, every enzyme works on a specific pH medium. Third one, activity of the enzyme according to the temperature. So temperature, the best temperature, we said that it's the normal body temperature. Yes, but the correct answer is 37 because the enzyme, they work at their best at the temperature which is 37 degrees Celsius. So the increase in temperature stimulates an increase in the activity of an enzyme after a certain level. But pay attention here. At temperature zero, you have to divide the graph into three different parts. First, at temperature zero, the activity of the enzyme was zero. As temperature increases from 0 to 37, the activity increases from 0 to 100. Then, as temperature increases from 37 to 60, this activity decreases back to 0. So the third property, every enzyme needs a proper temperature to act. Who can summarize the three properties for the enzymes right now? We have three important properties for the normal functioning of the enzymes. Who can summarize these three properties? Who can tell me the three properties for the normal functioning of the enzymes? Yes, Adam.
Okay, first one,
Yes,
the optimum temperature in this case study
Adam.
is
I'm talking
37.
Adam, I'm talking about the proper conditions for the normal functioning of the enzyme. What are the conditions needed for the functioning of the enzyme? I'm not talking about this slide. What are
Sorry.
the conditions needed for
فكرت
the
على رجلك.
functioning of the enzyme? Yes, Karim.
Ustaz, every enzyme needs a specific temperature,
Very good. Second one.
specific substrate, and
Very good. Third.
specific temperature, and the last specific substrate.
Specific temperature, specific substrate, and we still have one. Specific temperature, specific substrate, and yes, Celine,
Specific medium.
specific pH, specific medium. Thank you. Thank you, Celine, and thank you, Karim. So every enzyme needs a specific substrate. Every enzyme needs a specific temperature, and every enzyme needs a specific pH or a specific chemical medium. So to keep in mind, what is the optimum temperature? It's 37. Why? Not because it's the normal body temperature now. We started with this. But now, 37, because the enzymatic activity is at its best at this temperature. The enzymatic activity is best at 37 degrees Celsius. Inactive temperature is 0 degrees Celsius. Why? 0 degrees because the enzyme is inactive at this temperature. So the enzyme is inactive. So from now on, whenever we talk about 0 degree and below 0, the enzyme is inactive, meaning that the enzyme is sleeping at 0 degree and below. However, 60 degrees and above, it's called the denaturation temperature. You have to know this word. From now on, I am going to focus on this word, denaturation. I don't want you to use destruction. I want you to use the denaturation temperature. Why? Because the enzyme is destroyed, or you can use another word which is called denatured. So you have to know both of them, denatured and destroyed. So the enzyme is destroyed or denatured at high temperature. So very high temperature, it destroys or it denatures the enzyme. So what happened if the enzyme was at cold temperature, meaning that temperature 0 and below, 0 or -5, -10, the enzyme is inactive. But if we replace the temperature into 37, it will work again. This means that the enzyme can work again if it's inactive, if we replace it at 37. But if the enzyme was placed at a very high temperature, what will happen with the enzyme? The enzyme will be denatured or destroyed, and it will not function again, even though we replace it at 37. Moving for this exercise. In this exercise, what do we have? We have in this exercise, consider the following experiment that aims to study the different enzymatic property. We use salivary amylase as an enzyme, as bread, as an example on starch. So in first tube, we have bread plus salivary amylase. Yes, Maya, I need you to interpret the first experiment. First of all, what is interpret, Maya, before we start?
Interpret is indication and
Analyze plus?
Analyze plus indication.
Okay, start. So after adding...
After adding bread with the enzyme.
You can add amylase to the bread. So after adding salivary amylase to the bread.
Salivary amylase to the bread where the temperature was.
And which tube? We have to mention the tube.
In tube A.
Okay, let's start from the beginning. After adding and continue.
In tube A.
Yes.
In tube A, after adding salivary amylase to the bread, where the temperature was 37 degrees Celsius,
yes,
at pH 7, which is neutral.
Okay. What happens after 60 minutes? After 60 minutes, we have 0%.
The bread was 0% after 60 minutes.
Here you did the analysis. Very good. So we did the analysis. The second step is the indication. Very good. This indicates
that digestion occurs in tube A.
And digestion occurs in tube A. Very good. Moving for tube B. Yes, Khadi.
Yes, Ustaz.
In tube B. Good morning, Habibi.
Yes, in tube B. Adding bread plus Pepsin enzyme.
Okay, Khadi, let's start first with the correct way. So you have to say after adding Pepsin to the bread. Okay, now the bread بيكون inside the tube, we add Pepsin to it. Okay, Khadi?
Okay.
Yeah, let's start.
After adding Pepsin to bread.
Yes.
In tube B, after adding Pepsin to bread with temperature 37.
Very good.
At pH 7, which is natural.
Neutral.
There is no digestion.
So there is 100% bread. This is the analysis. Indication. This indicates that there is no digestion.
This indicates that occurs in tube B.
That no digestion occurs in tube B. Very good. Thank you, Khadi. Let's move now to new volunteers. I need the new volunteers. Let's move to Gabriel Hosseini, tube C. Interpret. Yes, interpret in tube C.
Tube C. After adding salivary amylase to bread,
yes,
in tube C with a temperature of 37 degrees and the pH of 2,
yes,
there was a positive iodine test.
Okay.
This indicates that no digestion occurred in tube C.
So positive iodine test meaning that we have starch, we mean that we have a bread, so there is no digestion occurs. Okay, thank you. Thank you, Gabriel. Thank you, Ghadi, and thank you, Maya. Moving for part 2. Specify the reasons why digestion does not occur in tube and the other tube study. So here digestion occurs where? Only in tube A we have digestion. In tube B and C we don't have digestion. They said specify. So whenever they said specify, what do we have to do? We have to justify. So here you have to justify. Moving forward, Ali Fadi Basma, justify why digestion does not occur in tube B.
Instead,
why digestion does not occur in tube B? Yes, Ali? Why we don't have digestion in tube B? Okay, thank you, Ali Fadi Basma. Let's move now to Hadil Sufan. Hadil, can you tell me why there is no digestion in tube B?
Yes, Ustaz.
Yes.
Um,
Yes, Hadil.
Ustaz, since the pH level, يعني the enzyme needs like a proper pH to act.
Okay, first of all, what is the enzyme here?
Pepsin.
Pepsin. It works on what, Pepsin? We studied this in the table. It works on what? This is a big problem. If we didn't study them, this is a big problem. Pepsin, it works on what? Yes, Hadil? Good. So how come we can solve such an exercise if we didn't study the enzymes that I told you hundreds of times about it? How come you don't have to study to answer such a question if you don't know what is pepsin and on what does it work? What is the substrate for pepsin? Let's see new volunteers here. Ismail Qurani, pepsin, it works on what?
Yes, Ustaz.
Pepsin, it works on what, pepsin? Good.
Mm.
Good, good, good. Good. By the way, in the exam, whenever you have an exam, you don't have time, for example, next time, you don't have time to look to the papers and to look to the handout and to the lecture to identify on which pepsin will act. Pepsin, it works on protein to give peptides. But here we have a bread. So it's not the specific substrate for it. So that's why there is no digestion. No digestion since pepsin doesn't work on bread. Pepsin, it works on protein. So we don't have protein here. While in tube C, we have bread plus salivary amylase. Temperature 37. Salivary amylase, it works on starch. Temperature is 37, but the pH here is 2. pH for the normal function of the salivary amylase is not 2, it's 7. So that's why it doesn't work here. So here, every enzyme needs a specific pH. While in tube B, every enzyme needs a specific substrate. That's why we don't have digestion. Those who didn't study the tables, I don't know what you are waiting for. I gave you about 2 to 3 weeks to study these tables and to review them because from now on, I'm going to work, all my lectures are going to be based on the tables. So if you didn't review the tables, this means that you are not going to be able to solve any of the exercises. Let's move now to the quiz that was so hard. Consider the following experiment that you use to identify studying the different enzymatic properties. So we use pepsin as a sample enzyme. I gave you a specific example. It's similar to the one we solved it, but I changed only the substrate and I changed the enzyme and some of the conditions. So here, tube A, we have albumin, that's pepsin. Pepsin is the enzyme that works on the protein. So we have temperature 37, pH equal to, which is acidic, 0% albumin. So interpret the experiment shown. I'm going to start in tube A. In tube A, after adding pepsin to albumin in tube A at temperature 37 degrees Celsius, where the pH is equal to 2, after 60 minutes, we have 0% albumin. What does this indicate? This indicates that we have digestion in tube A. Tube B, we have albumin plus pepsin. Temperature 37. pH, it's equal to 7. And the aspect after 60 minutes, it's 100% albumin. So here, the pepsin, it works on protein, which is the albumin. So here, everything is good. Temperature 37, also it's good. pH is 7, it's in neutral. But pepsin, it works in acidic, not in neutral. That's why we don't have digestion. So this indicates that there is no digestion in tube B. In tube C, we have albumin plus pepsin. Temperature 0, pH equal to, which is acidic. We have positive Biuret test. This indicates that the albumin is not digested. However, in tube D, we have starch plus pepsin. Temperature 37 and the pH equal to. So we have positive iodine test, meaning that the starch persists, is not digested. This indicates that the starch is not digested. Why the starch is not digested? Because pepsin is the enzyme that works on a protein and not on a starch. So sure, the starch will not be digested in the presence of pepsin. So here I did the interpret, and also I explained the reasons that prevent the digestion in the other tubes. So here, this is the quiz. The quiz was very simple, and it's very simple and similar to the one that we solved before. If you didn't study the notes, and if you didn't study the enzymes and the table of enzymes and the substrates, and you didn't review the food test, next quiz, you will not be able to solve any part of the quiz, because next quiz is going to be harder for sure. I'm going to go to level 2. This is level 1. This is a simple level. Next quiz will be level 2. So level 2 is going to be harder. So if you didn't review the notes, this means that you are not able to solve a question next time. So this means that the quiz is going to be so hard. So please, you have to review the notes regarding the enzymes, every enzyme, the substrate that work on, the product, and the temperature as well. And you have to review also the notes related to the food test, positive test, the negative results, and the positive results. You have to review also what is the temperature 0. It's called the inactive temperature, because the enzyme will be sleeping, will be inactive at this temperature. However, the temperature 60 and above, high temperature, it causes denaturation and destruction of the enzyme. And you have to know that when the enzyme is placed at the inactive temperature, when it's placed back at a good temperature, which is 37, it will work again. While if the enzyme was placed at temperature 60 and then we place it at temperature 37, the enzyme will not work anymore. So this means that the enzyme can function again only if it was inactive and not when it's destroyed. When the enzyme is destroyed and then placed again at 37, it won't function again. So I will repeat, this is the last time we're going to make revision. So mostly the chapter of digestion is almost done. So we finish about 90% of the chapter. Next time, it will be dedicated for exercises. So I'm going to make applications for this chapter. And also be ready to prepare yourself from next time. We may use graph paper. So we may use a graph. So we may draw a graph. So prepare yourself, prepare your graph papers for the next time, because next time maybe we are going to use graph papers in order to draw a graph and to analyze, to interpret, and to make, for example, variable factor and so on. So from next time, prepare yourself. We are going to do analysis, interpret, and anyone you didn't study yet the notes regarding the enzymes and the food test, you have to do so, because Enzymes are very important to solve the exercise. And you saw yourself. Some of you, they didn't study them yet. They used to open the notes and use the notes in order to solve. Next time, and the quiz is going to be longer. So I'm going to give you a bigger question. The question will be more in parts. For example, it's going to be 6 to 7 parts, and you have 40 minutes to solve it. So if you didn't study the notes, you are not going to be able to solve it. So pay attention to this note. This is the last time we're going to talk about the food test and the enzymes because they are very important to study them. Yes, Hadil, do you have a question?
Does cap normal box paper be like graph paper?
Box paper, it
Or
can
do we
be
like
like,
buy graph paper?
graph paper is different totally from the box paper. Box papers, every box is not 1 centimeter. Why the graph paper, every box is 1 centimeter. That's why, because you are going to be used to use this. And the next year you are going to use it a lot. Also this year you are going to use it in other subjects. So Hadi, graph paper is important. You have to know how to draw on the graph paper and you have to know how to for example, to use the graph paper. So I prefer to use the graph paper. So from next week, starting from the next week, I prefer all of you to have graph paper. And you have to
Yeah.
use it, and you have to use it in other subjects, not only in the biology. You're going to use it in math, also in physics, in chemistry as well. Yes, Ghadi, your question is?
It says
Yes,
I have two questions.
yes. First one.
The first one it says أنا الكويز عامل إنه عامله مثل ما هلا أنا كنت عم حل معك
Yes,
بس ما بعرف لأ أخدت 7 over 10 يمكن عندي كتير wrong words أو شيء.
7 over. It's over 5. First of all, it's over 5, not over 10.
Not
It's
over
over.
10?
It's over 5. So first of all, you did maybe so you didn't justify your reason. I think in the second part, and in the first part you have some mistakes. You remember exactly what are the mistakes. So we solve it right now. No problem. Next time you can fix these problems, buddy. Okay? This is the first question. Second question.
في امتحانات آخر السنة؟
Till now, I don't know. I can't answer this. So till now, we don't have. We don't have an answer for this. Okay? Yes, Gadi, your
Okay,
question.
thank you.
Sorry. Thank you, Gadi. Yes, Gabriel, your question is.
This was my mistake in the quiz because I got 4.5.
Your mistake, I think you didn't justify for the temperature. When the temperature was 0, you didn't mention that this is the inactive temperature. That's why you miss half. You didn't mention that 0 is the inactive temperature and the enzyme will be inactive. That's why I think. So to get four and a
Oh,
half,
that's why I got
that's
4.5
what you
too.
get also 4.5. Those who got 4.5, they missed this. They didn't mention that the inactive temperature is 0, so the enzyme will be inactive. Okay, that's why.
There's another question.
Yes, yep.
Do we have to
Excuse me,
send you the
Ustaz.
worksheet today?
I didn't hear you, Gabriel. Please, whenever one is speaking, the other should raise the hand. Whenever your friend finish, you can ask your question. Yes, Gabriel.
Do we have to send you the worksheet
No, no,
because
no,
we
no need,
already
no need, no need. Already you solved it the last week, so we solved it together this week. So no need to send it. Okay.
Mhm.
Okay, great. So other questions? Karim, you are as well asking question?
No خلص أستاذ. Thank you.
Okay, great. So questions? Others? Questions? Okay. Starting from the next week, pay attention to this. Next week, the quiz is going to be complex one. مش كتير صعب بس مش مثل هيدا. It's going to be harder. Why? Because we are going to move the second level, which is level 2. So level 2, the question is going to be harder. So I'm going to make it a harder question. So a complicated question. So هنبلش step by step. That's why I am telling you from now on for the 10th time, I think, that you have to study the notes related to the digestion notes, all the notes of digestion. Review them during this vacation because next time or next week, the quiz is going to be complicated because I'm going to move to the level, higher level. يعني ما حنضلنا questions simple مثل هيدا ال question. So we're going to move to the complex one. Yes, Karim.
أستاذ
Karim, I
important
didn't hear
thing
you. I
إن
didn't
يكون في
know
عنا
you.
وقت زايد.
I didn't hear you.
أستاذ المهم يكون في عنا وقت زايد.
No, ما في وقت زايد. So to grade 8 ما في 45 minutes exactly the quiz will be closed. It's enough for one question. And next year we will have 40 questions and you will have 1 hour. And I'm uploading all the questions and I'm شوف كريم هنحط لكم السوليوشن to upload your submission in the form of images. So it's much easier for you to upload your solution in the form of images and not in the form of PDF. And some of you, they insist to upload it in the form PDF. And I told you in the question, upload your solution in the form of image. And they insist, but don't PDF. I don't know why. So I'm putting images. So it takes about 5 minutes to upload. And the question needs 40 minutes exactly. Any question بالبيولوجي كريم in grade 8 and 9 ما بده أكتر من 40 minutes. So أنا بعطيكم وقت 40 minutes. From now on next week هيكون 45 minutes. 40 to solve the question and 5 minutes to solve
أستاذ
to
عامر
upload.
that's enough.
It's enough. It's enough. 40 minutes, it's enough. This question that we have it in this quiz, Kareem, it needs 40 minutes. This one, it needs 40 minutes. Do you think it needs 40 minutes? This question, if you study the notes,
No.
and you can solve it totally in 25 minutes. But others, they don't have time. They have time to chat and to send some picture. In this quiz, they didn't have time for this, and I sent them a warning for this. If you want to chat, if you want to cheat, so you're going to have 0. So some of them, they didn't get any of these marks, and then they're not working because next year, this lesson is exactly the first chapter in grade 9. So we're going to work in a similar chapter in grade 9. نفس chapter. Who is that? We're going to start it in grade 9. So if you study this in grade 8, it will be so much easier in grade 9. So now, if you have any question, I'm ready to answer before we end our session for today. Any questions? Some of you, I didn't hear the sound anymore. Amjad Ghanawi, can you hear me? Amjad Ghanawi? Amjad Ghanawi is out of service. Alma, do you have any question?
No.
Okay. Ali Fadi Basma, any question?
No.
Okay, great. Abbas, I didn't hear your voice today. Always Abbas was the first one to raise his hand. Today is sleepy right now. Abbas, good morning.
Yes.
Do you have any question?
No, I understood the lesson pretty good.
Okay, great, great. So Ismail,
Yes, أستاذ السمستر how much month؟ كم شهر؟
semester, how many weeks? مش how many months. How many weeks?
How many?
يعني, it's about 7 to 8 weeks, I think. 7 to 8
Okay.
weeks.
أستاذ
Yes.
أستاذ نحن بعدلنا بس لويك 40 حاطين على الآب إنه سكول يعني
لا
بعدلنا
لا لا
9
بعد
weeks.
في بعد weeks بعد في weeks بعد في شي 10 weeks تقريبا شي 10 weeks or
هو
11
9
weeks
weeks.
يعني 40 or 41 it depends حسب العطل and so on. Okay?
Just
So
can we leave?
can we leave? Yes, we can leave. Here we finish the session. Thank you all for joining this session and see you next week.
Thank you.